Hello everyone and namaskar. This is Chapter 13, Part 2, Photosynthesis, Part 2 of this chapter. It includes photopigments. It will discuss different types of photopigments and how can we separate these photopigments, that is chromatography. Then we have to see absorption and action spectrum of photosynthesis. And then we have to start mechanism, that is photosystem 1, photosystem 2. And then we have to say non-cyclic photophosphorylation. That is how the electron is being transferred in a non-cyclic way, and then splitting of water in this. Chapter 13, Part 2. This is Part 2, Photosynthesis in higher plants. In this part, we will see first photosynthetic pigments in higher plants. Photosynthetic pigments present in leaves of higher plants or any other green part of the plants. We can separate these pigments by chromatography, a technique to be used to separate these photopigments. Chromatographic separation of the leaf pigment, it shows the color that we see in leaves is not due to a single pigment, but the four pigments are there, and these pigments are chlorophyll A, which is brighter blue-green in color in chromatogram, chlorophyll B, that is yellow-green, xanthophylls, this one is yellow, and the carotenoids, these are yellow to orange. I repeat that there are four types of pigments, and these four pigments, one is that is chlorophyll A, which is bright or blue-green. This is chlorophyll B, number 2, yellow-green, xanthophyll, this one is yellow, and carotenoid, that is number 4th one, that is yellow to orange. So we have to see how we'll be able to separate, but before this we have to see detail of one by one. That is, you can see the same thing what I told you. That is, in a pestle and mortar, we have to take a leaf here, we'll keep some scent here, and with the help of that is mortar, we will crush the leaves so that the photopigment will come here in this. In this we have to add, that is acetone. Acetone will dissolve all these photopigments. Then we will filter, and we will have in a test tube, we will have this pigment solution. Now what we will do, we will take this capillary, one by one the drop will be kept here. The process is known as, that is spotting. If you have done in practical here, this is known as spotting. About 300 to 400 drops will be kept here on this spot. The pigments will be concentrated here. Then this chromatographic paper will be put here. In this is the chromatographic. This is chromatographic chamber. And in this chromatographic chamber, here we have the chromatographic solvent. And the lower end of this chromatographic paper will be dipped here in this. What will be now, this is already saturated one. So this solvent that will rise in this paper with the help of this capillary force, and from this spot that the pigments will get dissolved in this solvent, and according to their solubility they will go up and up. A beautiful color diagram we will have here. At the top we will have carotene pigment, then we will have xanthophyll here, then we will have chlorophyll A, then we will have chlorophyll B. It's very important, their sequence and their color you have to remember. Now we have to see detail of this chlorophyll. Chlorophyll is a tadpole-like structure. This is a tadpole-like structure like this. It is having a head. Head is known as porphyrin head, and it is having, that is 4 units, so called 1, 2, 3, 4. That's why this head is known as, this is porphyrin, or we can call that this is tetrapyrrolic head. This is tetra, that is pyrrole head. Is it? Chlorophyll A is the most abundant photosynthetic pigment. You have to remember, it is found in all, that is higher plants doing photosynthesis. It's the only pigment found in all photosynthetic plant. Important point. Chlorophyll A, it absorbs light maximum near to both ends of the visible spectrum, that is blue and red. It transmits or reflects green light. Therefore, they appear to be green, important one, while leaf that is appears to be green because the green light will be reflected by chlorophyll. And chlorophyll B, it is accessory pigment of that is light harvesting complex. Anyway, we were discussing about this chlorophyll. This is the head, and here we have a tail. That is, this tail is also known as that is phytol, and the overall structure of chlorophyll A is that is a tadpole-like. Is it clear? This chromatographic I told you, this fascinating one. In our school days, you can remember this practical, such colorful strip that one has to paste in practical records. Now you see here, that is carotenoids. These are yellow to orange pigment which absorbs light strongly in blue-violet region. These are known as shield pigment because they protect the chlorophyll from photooxidation or photobleaching. Light, when it is of high intensity, and also from oxygen produced during photosynthesis, that is carotenoids, they protect the chlorophyll. Therefore known as that is shield pigment. These are shield pigment. They absorb light and transfer it to chlorophyll for use in photosynthesis. As I told you, carotenoids are of two types. One is carotene, one is xanthophyll. Carotene is C40H56 hydrocarbons, most of them they being that is tetraterpenes, and the color varies from yellow to orange. Xanthophyll, oxygen is being added to this carotene, that is very similar to carotene, contains oxygen, and the color is yellow. We can remember here that is features, color, empirical formula, and the wavelength which they absorb to the maximum. Chlorophyll A, I told you this is blue-green in color. Empirical formula is C55 at 72 O5 N4 Mg, and it absorbs light at 430, and that is 662 nanometer, that is in blue region, and that will be that is in red region. If you say chlorophyll B, the color is yellow green. Empirical formula is C55H70 at 06 N4 Mg. The important point here is, what's the difference here in this? Here you see that is this has this methyl group, and here in this case the methyl group is being substituted by that is aldehyde group. This is the difference. Otherwise the rest of the structure is same. Come to absorption spectrum and action spectrum. What we mean by. Before we see detail, I'd like to that is make you acquainted with the basic experiment, the theme, the basic theme of the experiment, how that was being done. You see here absorption, what we mean by. First you see absorption spectrum is a graph showing amount of light of different wavelength absorbed by a pigment. You see here, let we have the pigments here. Let we are taking, for example, let this is chlorophyll A in one tube. In other tube we have, let this one is that is chlorophyll B. So what is being done? That is, this is the prism. The light will be splitted up, and here a monochromatic light will be given. Monochromatic light that is will be given. Let this is that is we have given violet. Let we have given blue. This color light one at one time will be given to chlorophyll A, and here we have an apparatus which can find out how much percentage of this that is wavelength have been absorbed by chlorophyll A. Did you get my point? So here we will have the data, that is how much violet have been absorbed by chlorophyll A, how much blue, what is the percentage absorption, and in that way the same thing will be repeated for chlorophyll B. Now you can come here, that you see this diagram. This is the wavelength of light in nanometer, and this is the absorption of light. Say for example that this is chlorophyll A. This is chlorophyll A. It has absorbed the light, maximum light in this region and then in this region. While this is chlorophyll B, it has absorbed the light of this region and here in this region. Two peaks we have here. So is it very clear to you? Absorption spectrum means the graph at that is one axis we have different wavelength of light and on other axis we have that is percentage of absorption by this particular pigment. Is it clear? The same thing will be done, that is action spectrum. Here simple we have to see that is here, let a plant is here. So just to make you understand that this plant has to do photosynthesis here. What will be done? In the same way that monochromatic light will be given to you at one time, one color, and we will see in this particular wavelength what's the rate of photosynthesis. Rate of photosynthesis. You know, rate of photosynthesis is measured, that is how much oxygen is being evolved. More oxygen is evolved, more rate of photosynthesis. So that will be known as action spectrum. What we mean by the graph, on one side we have wavelength of light, same thing here, graph showing rate of process of photosynthesis at different wavelength of light. Is it clear? Very, very important for examination point of view. Absorption spectrum, the graph which shows relationship between light of different wavelength absorbed by a pigment. That is on one axis, y-axis we have, that is here absorption of light, and on this axis we have wavelength of light. If you put this graph on this, that is what we'll see here. You see here is the peak here. Here is the peak here. If we'll keep absorption spectrum on action spectrum, then we found a close similarity between action spectrum and absorption spectrum of photosynthesis, and it shows that chlorophylls are responsible for absorption of light, that is blue and red, in photosynthesis. Is it very much clear to you? Yes, we will switch on to the next now. Mechanism of photosynthesis. The overall process of photosynthesis has two phases. One is light reaction, that is that depends upon light, and that is light, it depends upon that is light, it absorbs light energy, and this is followed by dark reaction. First light reaction, it is being followed by dark reaction. Light reaction we see, it is also called photochemical phase. What is there in this? Roughly speaking, you see here, let this is the chloroplast, let this is the leaf here, you know. In leaf, that this is the cell mesophyll cell, this is the cell mesophyll cell, this is the nucleus. Let here is the chloroplast, and this chloroplast we have taken out here. This is the chloroplast, just to see the structure. So in chloroplast, you know that this one is the granum. In granum we have the chlorophyll. So what is light reaction? On chlorophyll the light will fall. Light is essential for photosynthesis. You see, much more detail doesn't come in examination. Basic things you have to care for, basic things. Chlorophyll is there, that is water will be splitted up here, and that is oxygen will be released, and as a result of which here that is ATP and that is NADPH will be formed. The overall process is called light reaction. Is it very much clear to you? Can you give answer of one question? You know, in photosynthesis 4 factors are essential. Very important question that is children used to do, that is blunder in this. That is 4 factors: light and carbon dioxide from outside, chlorophyll and water, that is internal factor, are required for photosynthesis. Can you tell me, out of these 4 factors, how many factors are utilized or needed for light reaction? How many are needed for light reaction? You see the diagram. Light reaction, one is light. Light is very much important. And the other one is the light will be trapped by chlorophyll. That's why the chlorophyll is essential. And that you have to see detail of this in time to come. Water is also essential. So out of the 4 factors, how many factors have been used in light reaction? That is, these are over 3 factors have been used. Come to this point now. It is called photochemical phase. Include light absorption, water is splitting, oxygen is evolved, and formation of high energy, that is chemical intermediates, that is ATP and NADPH2. Here water will be splitted up, as a result of which oxygen is released. So pigments, these pigments herein, that is grainum, these are organized into 2 discrete photochemical light harvesting complex, photosystem 1 PS1, and that is photosystem 2, that is PS2. See, very important one, this one is the pigments present in grainum. We can group them into photosystem, that is first, and photosystem 2. This is again important one. One is first, one is second. One may be confused that the first is important, discovered first, second is less important. It's not like this. They are named in the sequence of their discovery. That is, this has been discovered first, not that this is very important. That's why this is called first. No, that is not in sequence in which they function. Light harvesting complex are made up of 100 of pigments molecule bound to protein. Each photosystem has all pigments except one molecule of chlorophyll, that is which make the, that is antennae of this. These pigments help to make photosynthesis more efficient, but by absorbing different wavelength of light. So here this is, that is light harvesting complex, this one is. These are the different photopigments, and here this is the reaction center, that is they will accept the photon energy, and ultimately all these will transfer to this reaction center. We can write it like this also. This is reaction center, and these are the, that is antennae molecule, and this one is, that is reaction center, right? Now PS1, the reaction center here is, that is an absorption peak 700, hence this is P700, and in pigment systems second we have reaction center 680. Some other differences we can see here, that is pigment system first or photosystem first, reaction center is P700. It lies on the surface of 𝕆𝕋𝔼. If I draw here, that is, this is the 𝕆𝕋𝔼, that is usually PS1 is on the surface here, while PS2 that lies, that is, it is non-oppressed surface, and this is on oppressed surface. Found in both grainum and stroma, participate in both cyclic as well as non-cyclic. We have to see detail of this, and they are not associated with the splitting of water. Very important points here. Photosystem 2, it occurs on inner side of the 𝕆𝕋𝔼. Let this is the 𝕆𝕋𝔼, children. Let this is the 𝕆𝕋𝔼. PS1 is present here. This is the lumen of 𝕆𝕋𝔼, and PS2 is found on inner side. It is involved only in a non-cyclic flow of electron and associated with, that is, splitting of water. Okay. Now you see electron transport system. What we mean by, you see, in photosynthesis light energy is to be trapped by using absorption of this light energy. The chlorophyll molecule, let this is the chlorophyll molecule. After absorption of that is energy, chlorophyll molecule will be excited. The electron will jump in outer orbit, and ultimately this chlorophyll will be oxidized here, and the electron that will come out from this. Now this electron, it goes through electron transport system, and it will lose its energy, and this energy will be utilized in the synthesis of ATP. We have to see detail of this. If the electron, after generation of ATP through electron transport system, is returned to the same chlorophyll molecule here, then children, that will be known as cyclic photophosphorylation. Phosphorylation means formation of ATP. And if this electron, after generation of that is ATP, it is not returned to the original chlorophyll molecule, it has gone somewhere else to that is NADPH, then that generation will be known as non-cyclic one. I may repeat it. Chlorophyll will be excited by using that is light. Is it clear? That is excited, that is normal state, or we may call that is reduced state or ground state, and this electron will come out here. Now see the basic things here. This is non-cyclic photophosphorylation, that is photosystem second. You see this, that is mark here. In non-cyclic photophosphorylation you are seeing photosystem second and photosystem first. Both are involved, as I told you in differences also. What will be now? That is here, this is the light harvesting complex of PS second. It has absorbed the light energy. Electron will come out here. This electron will be accepted by electron acceptor. Then from this electron acceptor, it will go to electron transport system. Then after this, that will go to the light harvesting complex of PS1. This is upward movement, downward, upward movement. Remember, now it will be accepted by electron acceptor of photosystem 1. And now this electron, after this it will come out, will be accepted by NADP. That is, the electron will also accept, that is proton will become hydrogen, and ultimately it will reduce this NADP. Is it clear? So you have seen in non-cyclic photophosphorylation, that is they will be responsible for ATP generation. How that will be, we have to see detail. And here formation of that is ATP, and then this is reduction of NADP to NADPH. Now the other important point here is, that is in this, the how the electron will come, will be back. This electron will be returned to the same chlorophyll molecule by photolysis of water. That is non-cyclic photophosphorylation. This is called JEE scheme. You are seeing that is known as JEE scheme. While in cyclic photophosphorylation, the electron, you see here, it has arisen, will go to the electron transport system and will be back. It will generate only ATP, and here NADPH2 will not be formed. That will release oxygen here. It will make, that is this one, and it will make ATP. Is it clear? Now we have to see detail of this. Non-cyclic photophosphorylation, come to this point. Photosystem second, you know, reaction center is this one, will absorb the wavelength of red light. Electron will be excited. They will jump into the higher orbit. I told you here, electrons will be picked up by electron. You see, that was the light harvesting complex. Electron have been received by here electron acceptor, is it? And then from here they have gone to the cytochrome. The movement of electron is downhill in terms of oxidation reduction potential. Electrons are not used as they pass through electron transport system, but they are passed to the pigment of photosystem. Here this is electron transport system, here photosystem first, and then from here they will go to this electron, that is acceptor, and now will be back to that, they will, that is reduce this NADP. The same thing I told you. Simultaneously, electron in the reaction center of PS1 also excited, receive red light, and then these are transferred to another acceptor molecule which is having greater redox potential. Is it clear? Now the electron, the repetition of the same what I told you, that is downhill movement again. That is from here, you see they have gone like this, they have gone to electron transport system, they have gone, this is PS1, and this was, that is PS, that is second. Now again they will go downward here, that is molecules, and they will make energy rich compound here. In addition of these electrons that will be formed, the whole sequence of the electron starting from this PS second to PS1, that is known as JEE scheme, and it is a characteristic shape because Z shape this is. See, splitting of water, they told you, that is in this photosystem second, that is the light has been absorbed, electron has come out. The electron, they will not be back to this chlorophyll molecule. So how this chlorophyll will come to the normal state? Here the water will be splitted up. The process is known as photolysis of water. This is photolysis of water, is it? And splitting of water is associated with this. Water is split like this. It creates oxygen, and net product of the photosynthesis. Here the oxygen will be released as a result of that is in photosystem second. It need to be emphasized here, water splitting complex is associated with PS second. Itself is physically located on inner side of the membrane of thylakoid. That is, this is water molecule. Protons will be formed, electrons will be formed. Electron will go to this, that is photosystem second, and oxygen is released. Is it clear? Now you can summarize what is therein. That is non-cyclic photophosphorylation. Can you tell me? There is formation of, that is ATP, there is formation of NADPH, and there is photolysis of water, as a result of which that is oxygen is released. This is part second. Thank you.